Embryonic stem cells are the master builder cells. They are pluripotent, meaning they can differentiate into any kind of cell in our body, such as muscle, nerve, intestinal, or cardiac cells. Embryonic stem cells also have the ability to replicate themselves indefinitely. Today, stem cells have widespread applications in research and regenerative medicine. Researchers are testing the effects of drugs on particular cells, as well as modelling pathogens. Stem cell donations are used to heal people or fight disease. They are also figuring out how to grow different tissues and organs in the lab. In future, it is hoped to assist in the treatment of diseases such as diabetes, Parkinson's, and heart disease. This study aims to solve the question of whether a cell's fate is predetermined or whether it could be influenced by an outside stimulus. It also aims to determine a time window at which a cell's fate is sealed by studying live stem cells throughout their cell cycle. OCT4 is a pluripotency factor in human embryonic stem cells. The lower the amount of OCT4 expressed, the more likely that cell will be to differentiate, while the higher the amount, the more likely it is for a cell to self-replicate. A red fluorescent marker was attached to the OCT4 in the live cells, so the expression could be observed in time-lapse before and after a differentiation signal, BMP4, was added. Once the fate of the cells were known, the ancestor cells were traced back, now knowing the fate of their offspring. Cells destined to self-renew had higher OCT4, frequent bursts of increased OCT4, and faster cell cycles. These were all observed as early as 2 days prior to the introduction of BMP4. The studied populations showed that OCT4 levels are passed from mother to daughter. Slightly different amounts of OCT4 are given to each daughter cell, which is how different fates can arise from the one population. See the difference between the first and the final generation? By looking at the mother's cell's OCT4 levels alone, it was possible to predict the daughter's fate with around 80% accuracy. The levels of OCT4 are determined within the first half hour of a cell's life, and within 6 hours it was possible to predict its fate with a whopping 93% accuracy. This study has shown that a cell's fate is almost entirely predetermined by maternal OCT4 expression, inherited OCT4, the frequency of bursts of OCT4, and the duration of the cell's cycle. External stimuli can only play a very minor role in this. A precise time window was also determined for fate prediction. In light of this, perhaps the concept of pluripotency should be rethought as a heritable trait rather than a single cell's ability.